Hello everybody. So now we are going to discuss smooth muscle. I told you already that features of smooth muscle are completely different from other two muscle, that is the cardiac muscle and skeletal muscle. So what are the special feature in smooth muscle? Number 1, smooth muscles are spindle shaped. It is spindle in shape, so spindle shaped single nucleated cell. Single nucleated cell, right? So we have seen in skeletal muscle that skeletal muscle is a longitudinal fiber with multiple nucleus, but smooth muscle cells are spindle in shape. So if this is a single smooth muscle cell, okay, single nucleus and spindle in shape, and suppose this is another smooth muscle cell, so spindle shape and single smooth muscle. Second point that you have to remember is that proper neuromuscular junction is not present in smooth muscle. Like for example, skeletal muscle, we have seen that there is a proper neuromuscular junction. But in smooth muscle, we all know that it is supplied by autonomic nervous system. So when the branches of the autonomic nervous system supplies the smooth muscle, it does not form a proper neuromuscular junction. Look at the diagram first, you will understand better. So suppose this is a branch of the autonomic nervous system. So this branch is coming, it is entering in between smooth muscle. Now in multiple areas, it is forming a spherical region. So this branch of the autonomic nervous system, maybe this is sympathetic or parasympathetic, it is giving supply to smooth muscle. But look at this supply. This is not a proper neuromuscular junction, but rather I can find out multiple spherical region here. This spherical region contain the neurotransmitter, okay, maybe acetylcholine, maybe epinephrine, maybe norepinephrine, whatever. This neurotransmitter is present within this spherical region, and this spherical region is known as varicosity. So this is known as varicosity, right? So this varicosity will release the neurotransmitter, and then that neurotransmitter will be spreaded throughout the surface membrane of this smooth muscle. That means I am not seeing any proper neuromuscular junction where neuron is touching the smooth muscle membrane, but rather there is multiple varicosities. So that's why I am writing here that autonomic nervous system forms multiple varicosities, and this is known as neuroeffector junction. This is known as neuroeffector junction rather than neuromuscular junction, right? So this is a new term given in Genome, the neuroeffector junction. And this neuroeffector junction, sometimes known as synaptic inpatient. Synaptic inpatient. What is the meaning of that? Meaning, as if when the nerve is passing through the muscle fiber, okay, look at this. This nerve is passing through the muscle fiber. During its journey, it is giving some supply to the muscle, giving some neurotransmitter to the muscle. This kind of synapse is known as synaptic inpatient, but the better term which is used nowadays is neuroeffector junction rather than neuromuscular junction in smooth muscle, right? So this is the second point. Now third point is that attachment between smooth muscle is done by two molecules. One is there will be presence of adherent junction. Okay, we have seen earlier what is adherent junction, and there may be presence of gap junction in between smooth muscle. So let me draw here. So in between two smooth muscle cell, there may be presence of channel protein, that is gap junction. So the gap junction channel protein may be present. Okay. Apart from this gap junction, two smooth muscle remain attached with one another with the help of the attachment junction, and this is known as the adherent junction. So this is adherent junction and this is gap junction, right? So because of the presence of gap junction, we know what is the property of gap junction. Through gap junction, small molecule like calcium, like sodium, they can pass through one cell to another cell. Due to the presence of this gap junction, smooth muscle acquire another property, and that is the property of syncytium property. What I'm saying, note down the fourth point. Type of smooth muscle. Type of smooth muscle. Okay. So there are two main types of smooth muscle: single unit smooth muscle, single unit smooth muscle, which is also known as unitary smooth muscle, and second one is the multi-unit smooth muscle, multi-unit smooth muscle or multi-unit muscle. Now the single unit smooth muscle means when all the smooth muscles are connected with one another by gap junction. Look at my diagram. So this is one smooth muscle, another smooth muscle, another. So multiple smooth muscle, multiple smooth muscle. What is the specialty is that all the smooth muscle, they are connected through this gap junction channel. So because of the presence of this gap junction channel in between them, what will happen whenever one neuron, suppose this is autonomic neuron, it is stimulating, giving stimulation here on a single muscle cell, the calcium will be released here, and that calcium will easily pass through one cell to other cell, one cell to other cell. And what is the net result? Net result is there is contraction of all of this smooth muscle simultaneously as an unit. So this simultaneous contraction property as an unit is known as syncytial property. You must have heard this word, syncytial property, right? So this property is also inherent to the cardiac muscle. We will be discussing later on. But syncytial smooth muscle means all the smooth muscles are connected with one another via gap junction, and due to which whenever there is contraction, they are contracting as a single unit. This is known as the single unit smooth muscle or unitary smooth muscle. Most of the smooth muscle in our body, if you look at the examples of this unitary smooth muscle, suppose gallbladder contraction. Whenever the gallbladder is contracting, it is contracting as a whole. So all smooth muscle of this gallbladder, they are contracting simultaneously because huge amount of gap junction is there. Suppose contraction of uterus, right? Okay, not only uterus, suppose contraction of the GIT. Large part of the duodenum is contracting. So what is happening? How the large part of the duodenum is contracting? Because multiple gap junction is connecting the duodenal smooth muscle. So all these gallbladder, ureter, uterus, okay, GIT, all these are examples of smooth muscle which are single unit or unitary smooth muscle. So unitary smooth muscle means the contraction will be gross contraction. Large part of the muscles is contracting simultaneously. But multi-unit smooth muscle means when the number of gap junctions are very, very less. Suppose this is one smooth muscle, this is another smooth muscle, and both of them are supplied by different branch of the nervous system. Suppose this is one nervous system, this is another neuron. Both of them are supplied by different nerve fiber. So what will happen? This green colored smooth muscle, they will contract as a single unit. Okay, that means within a smooth muscle, there are multiple unit because the gap junction are not connecting these smooth muscles with one another. This is typically known as multi-unit smooth muscle. Multi-unit smooth muscles are required for fine control. Whenever you require fine control, you require multi-unit smooth muscle. So examples of this multi-unit smooth muscle is iris muscle, okay? So the muscles of the iris, okay? Ciliary muscles of eye, as well as the piloerector muscle, piloerector muscles of the skin. All of them are examples of multi-unit smooth muscle. Fine adjustment is done by the contraction of the smooth muscle, and I can understand that multi-unit smooth muscle means gap junctions are very, very less, or there may be no gap junction at all. But single unit smooth muscle means gap junctions number are very, very high, and that's why they are behaving like a single unit. So getting my point? So this is the importance of presence of gap junction in smooth muscle, right? Now next we are going to understand that the structural part of the smooth muscle. So if we look at the smooth muscle cell, I already told you they are spindle in shape. Now I am asking myself, is there any presence of T-tubule on smooth muscle membrane? Answer is T-tubules is absent. T-tubules in smooth muscle is absent. But rather than T-tubule, there is a small depressed area on the surface of the smooth muscle membrane like this, okay? So suppose this is a spindle shaped smooth muscle cell. There will be a little depressed area. This is not a complete tubules, but only a depressed area. This depressed part, it is said that this is a rudimentary T tubule basically on smooth muscle. The name of this depressed part is known as CAVE, caveoli. CAVEOLI, caveoli. What is the importance of caveoli? At the level of caveoli, there will be presence of multiple voltage gated calcium channel. We have also seen in skeletal muscle and cardiac muscle that T tubules contain voltage gated calcium channel, L-type voltage gated calcium channel. Same for the smooth muscle, but here it is not T tubule, here it is known as caveoli. And caveoli is the area where multiple L-type voltage gated calcium channels are present. Okay, so T tubules are absent, but in presence of T tubules, the caveoli are present, right? Okay, next I am asking, actin myosin, is it present or not? Obviously. Okay, so actin and myosin, are they present in case of smooth muscle? Obviously it is present, but the actin myosin are not highly organized. So I am writing here, although it is present, but it is not highly organized. Okay, I will tell you what is the meaning. Not highly organized. Some of the actin myosin, they remain obliquely within the muscle. So if you look at this diagram here in the smooth muscle, look at, I am drawing actin and myosin. So one point from here, actins are radiating, another point here on the membrane, from there multiple actins are radiating. So this red color one, which are radiating from a point, these are actin, and in between this actin there will be presence of myosin, right? So this is one sarcomere. Now if you look at the another sarcomere, then you will find out that the direction of the actin is not in the same direction. These actins are present obliquely, right? So in between there is myosin. So if we look at the sarcomere in the smooth muscle, then it is not well organized like that of the skeletal and cardiac muscle. It is not parallel to one another. It is not well organized. Some of the sarcomere is present obliquely. Because of this, you are not going to find out alternate dark and light band within the smooth muscle. Rather, we say that smooth muscle is non-striated muscle. So this is a non-striated, okay, muscle, because you are not going to get alternate dark and light band because of this disorganized actin myosin complex, right? Now you look at, in cardiac and skeletal muscle, we have seen that the actin are parallel to one another, right? You know, so these actins will be like this, and in between myosin will be there. But here what you are finding out, all the actins are radiating from a single point. This single point where all the actins are attached, this is known as the dense body. If I ask you, actins remain attached to it, which line in case of skeletal muscle? All of you say that this is known as Z line in case of skeletal muscle. So are you getting any line here or any disc here in case of smooth muscle? Answer is not. So the next point that you have to remember, point number 7, that there is absent, okay, absent Z line or Z disc. Rather than Z disc, there is a spherical portion which is known as dense body. Okay, so please remember, okay, in case of smooth muscle, T tubules is absent, but homologous organ of T tubules is caveoli. Z line is absent, but homologous part of Z line is dense body, right? Then next I am asking, actin myosin is done. Now I am asking that what about troponin and tropomyosin? Very, very important. Is troponin is present in smooth muscle? Answer is troponin is also absent in smooth muscle. So troponin was the calcium binding protein in cardiac muscle and skeletal muscle. If calcium binding protein is absent in the smooth muscle, so you must have to give another calcium binding protein, and that is known as calmodulin. Very, very important. So the calcium binding protein in case of smooth muscle is calmodulin. Multiple times this question has been asked in MCQ. So calmodulin, you cannot forget. Calmodulin is the calcium binding protein in case of smooth muscle, right? So calmodulin is present, but troponin is absent. Now, next is, what about tropomyosin? What about tropomyosin? Is it present or absent? Answer is yes, it is present. Tropomyosin is present in smooth muscle. What was the function of tropomyosin in skeletal muscle? It was preventing actin-myosin interaction. But look at, in case of smooth muscle, tropomyosin is present and it enhances actin-myosin interaction. It enhances the actin-myosin interaction. Oh my God. Okay, so it is increasing the actin-myosin interaction rather than preventing. So question is, then who will prevent the actin-myosin interaction in smooth muscle in resting condition? Look at two new names you have to remember. This is important for future MCQ. Calponin protein, as well as caldesmon protein. These are the two proteins which prevent the actin-myosin interaction in smooth muscle. It prevent, okay, actin-myosin interaction in resting condition of smooth muscle. So this is protein which is specially present in case of smooth muscle, prevent actin-myosin interaction. Calponin and caldesmon tonically inhibit actin-myosin interaction, right? So calponin and caldesmon is done. And number 11 is nebulin and titin. What about these two proteins? Titin and nebulin, they are also absent in case of smooth muscle, right? So many features we have seen. We have seen the caveoli, we have seen dense body, we have seen calmodulin, and we have seen this calponin, caldesmon. They are the very new names, particularly to the smooth muscle, and we have seen the certain structure are absent in smooth muscle in comparison to skeletal muscle. Now, apart from these features, there are also difference in mechanism of contraction in case of smooth muscle. So now we are going to understand the mechanism of smooth muscle contraction, but before that we have to understand that what are the sources of calcium. Sources of calcium for smooth muscle contraction, right? So skeletal muscle we know, cardiac muscle we know. What about smooth muscle? What is the source? So I am drawing the smooth muscle membrane here. Okay, so this is the smooth muscle membrane, right? So I hope you understood that which part is the caveoli. So at the level of this caveoli, I told you already that there will be presence of voltage gated L-type calcium channel. So this is L-type voltage gated calcium channel, right? This is calcium channel. So first of all, whenever there is presence of action potential on the surface of this smooth muscle, the same phenomena like that of the cardiac and skeletal muscle, the action potential will travel down, right? So this will causes depolarization of the smooth muscle membrane, and this depolarization is going to activate this voltage gated calcium channel. So it is activating the voltage gated calcium channel. If I ask you, could you recall which subtype of voltage gated calcium channel this is? You obviously answered that, sir, this is CAV 1.2 type of calcium channel. I told you repeatedly that CAV 1.1 is for skeletal muscle and 1.2 is for cardiac muscle as well as smooth muscle. So whenever there is depolarization, this calcium channel will open up, and through which extracellular calcium is going to enter within the smooth muscle. So this calcium is coming from extracellular fluid, right? Now, in case of smooth muscle, the sarcoplasmic reticulum is present, but that is rudimentary. So I am drawing a sarcoplasmic reticulum. So this is sarcoplasmic reticulum, but this is not like that of the skeletal muscle sarcoplasmic reticulum. In skeletal muscle sarcoplasmic reticulum, we have seen two spherical region that is known as the cisterna, and that was storing huge amount of calcium. But here we are not going to get that. Here is a small sarcoplasmic reticulum, rudimentary sarcoplasmic reticulum. But at the level of this sarcoplasmic reticulum, there will be presence of channels, calcium release channel, and you know the name of this channel. This is RYR type 3 channel. So, whenever the calcium is entering from extracellular fluid through this voltage gated calcium channel, this calcium is going to stimulate this RYR type 3 channel. What type of mechanism this is? So outside calcium is stimulating RYR type 3 channel, and through this RYR type 3 channel more calcium will be released into the cytoplasm. This phenomenon we have already seen in cardiac muscle, and this is known as CICR phenomena, calcium induced calcium release phenomena. We have seen already calcium, outside calcium is inducing release of intracellular calcium, that is RYR3 type channel. So this is known as calcium induced calcium release. So far, if I write down that what are the source of calcium, if I answer this question, so first I am writing number 1 source is ECF calcium, which is entering inside the smooth muscle cell via CAV 1.2 channel, right? Second source I am writing sarcoplasmic reticulum calcium, which is released via CICR mechanism, right? Now, on the smooth muscle surface, there are many receptors. This mechanism is very separate from other mechanism. There are many receptors. This receptor are receptor for various hormones, neurotransmitter, okay, etc. So maybe drugs, okay, maybe hormones. So it's maybe various drug molecule. It may be acetylcholine molecule or norepinephrine molecule, or it may be different hormones. So there are various receptors on the surface of this smooth muscle cell. Now, sometimes if you activate this receptor, there will be production of second messenger, and one of the second messenger system we know that is going to produce DAG and IP3. Okay, so this second messenger system we are going to discuss in endocrine physiology, but for the timing remember that whenever you are stimulating this receptor, okay, it's a G protein coupled receptor, it is producing a second messenger, and the second messenger system is generating inositol triphosphate and diacyl glycerol. Now this inositol triphosphate can also release sarcoplasmic calcium. How? At the level of this sarcoplasmic reticulum, there is a receptor for this inositol triphosphate, and this receptor is a calcium channel. So what I am saying, this is IP3 receptor. This receptor itself is a channel. So whenever the IP3 is produced inside the cell, this IP3 is going to stimulate this receptor, okay, and because of this stimulation, the channel or the receptor will open up and further calcium will enter in the cytoplasm. So the third source of calcium I can write down here, that this is also sarcoplasmic reticulum calcium, but it is coming via IP3 receptor, inositol triphosphate receptor. So RYR type 3, that is also releasing calcium, and inositol triphosphate 3, that is also releasing calcium. Very, very important. This question has been asked multiple times, that IP3 is releasing calcium in the smooth muscle, which we have not seen in case of cardiac muscle or skeletal muscle. Now, apart from all this mechanism, there is a fourth mechanism, and that fourth mechanism is known as SOCE, store operated calcium entry. Look at what I am saying. On the surface of this smooth muscle, okay, on the surface of this smooth muscle, there is a channel. There is a channel. This channel will open up whenever intracellular store of calcium is less. So whenever your cell calcium is less, it will send some signal to this surface calcium channel by some unknown mechanism which we do not know. But this mechanism is very, very special to certain smooth muscle whenever intracellular calcium store is depleted, right? or this sarcoplasmic reticulum store of calcium is depleted, it will send signal to this surface calcium channel, and this calcium channel will open up, through which more calcium will enter, and this calcium will replace the sarcoplasmic store and cytoplasmic store. This channel is known as store operated calcium entry or store operated calcium release. So SOCE means, you remember, this is nothing but store operated. This is store operated calcium entry into the cell. So now I can understand that there are four major mechanisms of calcium, source of calcium in smooth muscle. Number one, ECF calcium is entering inside the cell via CAB 1.2. Sarcoplasmic calcium is entering via CICR, that is calcium induced calcium release mechanism. Sarcoplasmic reticulum is also entering via IP3 receptor, and there is another one, that is store operated calcium release. Now, if I ask you that this third and fourth mechanism, that is inositol triphosphate is releasing calcium, look at this diagram. So this inositol triphosphate, that is releasing calcium, and the store operated channel, that is allowing calcium to enter in the cell. Do you require action potential or do you require depolarization for this calcium release? Answer is no. So what I am saying, this IP3 and SOCE, these two mechanisms, it releases calcium which is independent of depolarization of the membrane or independent of action potential on cell. Why I am saying this? Because in skeletal muscle and in cardiac muscle, without action potential, without depolarization, you cannot cause contraction of the muscle. But in case of smooth muscle, if you give some drug, so here I told you that if you give some drug or acetylcholine or some hormone, okay, so they are going to stimulate this channel. So this drug, hormone, they are going to stimulate this channel, and if you stimulate this channel, it will produce IP3. IP3 will stimulate its receptor, and because of this receptor stimulation, calcium will enter in the cell. That can cause contraction of the muscle without generating action potential, without generating depolarization of the membrane. Very, very peculiar for smooth muscle. That's why you have to remember that in case of certain pharmacological agent, in case of certain drugs, the contraction of the smooth muscle is possible without any depolarization. This is known as pharmacomechanical coupling. This is known as pharmacomechanical coupling of smooth muscle. Getting my point? So we have seen electromechanical coupling in case of skeletal muscle, electrochemical coupling in case of cardiac muscle, and now we are showing pharmacomechanical coupling in smooth muscle in presence of certain pharmacological agent, like for example drugs or acetylcholine or hormone. There is mechanical contraction of the smooth muscle without any depolarization, without any action potential. This is known as pharmacomechanical coupling of smooth muscle, right? So now we understood that what are the different sources of calcium within the smooth muscle. Now after this, we are going to discuss the mechanism of smooth muscle contraction. The mechanism of smooth muscle contraction. Smooth muscle contraction. Very, very simple. Okay. So again, the contraction will be based on calcium only. So calcium inside cytoplasm of smooth muscle, right? So what this calcium is going to do? It is going to cause contraction, but it has to bind with some molecule, but troponin is absent. So who is going to bind with this calcium? So calcium binds with calmodulin, very, very important, calmodulin. Now it will form a calcium calmodulin complex, calcium calmodulin complex, obviously. Okay. Now this calcium calmodulin complex is going to activate enzymes, various enzymes, kinases, right? So I'm writing here, it is activating calcium calmodulin complex is activating myosin light chain kinases. Okay, myosin light chain kinase. So MLCK is activated. So this is an enzyme. What is the importance of this enzyme? Where this myosin light chain enzyme is present? It is present there in the cell, right? So this myosin light chain kinase is now going to activate, or it is going to causes phosphorylation of, okay, phosphorylation of myosin light chain. Getting my point? Myosin light chain. Where this myosin light chain is present? Look at this diagram. Okay, so this is myosin, like that of the skeletal muscle or cardiac muscle, and I told you it contains tail region, neck region, and a head region like this, right? I told you already in school. So this is type 2 myosin. Okay, so at the level of this myosin head, there are presence of certain light chain. So myosin head is composed of light chain and heavy chain. Light chain, one of the light chain is known as regulatory light chain. That light chain phosphorylation is essential for smooth muscle contraction to start. Now question, why? Okay, is this myosin light chain is also present in skeletal muscle myosin and cardiac muscle myosin? Answer is yes. But we have not discussed about phosphorylation of this myosin light chain there in smooth muscle, cardiac muscle or skeletal muscle. Answer is yes. We have not discussed because that is not required in skeletal muscle and cardiac muscle contraction. But phosphorylation of this myosin light chain is essential for smooth muscle contraction. Why? Whenever you phosphorylate this, there will be increase in the angle of this myosin head. What do you mean by that? Means normally myosin head remain at a 90-degree angle in case of skeletal and cardiac muscle, but in smooth muscle it remain in a very lesser angle. So I have to increase the angle of the myosin head to start the contraction. Second thing, at the level of myosin head, there is a presence of an enzyme, that is myosin ATPase enzyme. This enzyme activity is very, very low in smooth muscle under resting condition. So I have to increase the myosin ATPase activity, and I also have to increase the head neck angle in case of myosin. So in case of myosin, head neck angle you have to increase, and you have to increase the myosin ATPase activity. In case of skeletal muscle, cardiac muscle, myosin ATPase activity is constitutively high. That's why you don't have to increase the activity of the myosin ATPase. And this angle is already high in skeletal muscle and cardiac muscle. So that's why this two is not required in skeletal muscle and cardiac muscle. That's why myosin light chain phosphorylation we have not discussed there. But in smooth muscle, we have to do these two things. So whenever you are causing phosphorylation of the myosin light chain, what is going to happen? Just now I told you that there will be increase angle of myosin heads, okay, and there will be increase myosin ATPase activity. Now just after increase of this angle and ATPase activity, now there will be formation of cross bridge formation, right? And after cross bridge formation, we know that there will be presence of cross bridge cycle generation. And cross bridge cycle generation means that is nothing but muscle contraction. So this cross bridge cycling is similar to that of the skeletal muscle and cardiac muscle. It will leads to muscle contraction. But hold on. So whenever you are increasing the myosin head angle, okay, increasing the myosin head angle, as well as you are increasing the myosin ATPase activity, there is actin myosin interaction, right? But you have forgot one thing, that is, what about calponin and caldesmon? What about calponin and caldesmon? Because you told earlier, sir, that calponin and caldesmon, they are very specific molecule which are present in smooth muscle which prevent actin-myosin interaction. So note down. Okay, I have to prevent that inhibition also. So whenever there is formation of calcium calmodulin complex, this complex also activate various other kinases, right? This kinases causes phosphorylation of this inhibitory protein, that is calponin and caldesmon. Because of the phosphorylation of calponin and caldesmon, what will happen? There will be prevention of inhibition between actin and myosin. Now, whenever this inhibition is removed, and whenever this myosin ATPase activity is high, and whenever this myosin head is already raised in angle, now all this will leads to cross bridge formation. So getting my point, how the smooth muscle contraction is happening? You have to cause phosphorylation of the myosin light chain, as well as you have to remove the inhibition. Removal of the inhibition of calponin and caldesmon is also done by phosphorylation, and phosphorylation of the myosin light chain is increasing the angle of myosin head, and that is causing cross bridge generation, and then cross bridge cycling is going on. Although I told you earlier that cross bridge cycle is similar to that of the skeletal muscle, but remember, the rate of cross bridge cycling in smooth muscle is 1/10 of skeletal muscle, rate. So what was the rate in case of skeletal muscle? I told you in case of skeletal muscle, the rate is 5 cross bridge cycling will occur per second. I hope you remember. In case of smooth muscle, this rate is very slow. This is 1/10 of skeletal muscle cross bridge cycling rate. Getting my point? This you have to remember. So now whenever muscle contraction is going on, how to causes relaxation of this smooth muscle? Means now you have to cause a relaxation of the smooth muscle. You know that there is another enzyme which is known as MLC, myosin light chain phosphatase, right? So whenever this myosin light chain phosphatase enzyme is activated, it will causes dephosphorylation. Of myosin light chain, right? Whenever myosin light chain is dephosphorylation, it will causes detachment of myosin heads from actin, right? And then if the calcium is already removed from the cytoplasm, then this will causes a relaxation of the smooth muscle. Now, in certain smooth muscle, there is a very peculiar property. So we have just seen that dephosphorylation, is causing detachment in certain smooth muscle. Even after dephosphorylation, the myosin head remain attached with actin, right? So dephosphorylation ideally should causes detachment, but I am saying that in certain smooth muscle, even after dephosphorylation, myosin heads remain tonically attached with actin, right? So I can understand that due to tonical attachment with actin, there will be a sustained contraction of the smooth muscle, although the myosin light chain is dephosphorylated. So this is a sustained tonic contractile state of smooth muscle. I can write down like this. This is nothing but a sustained tonic contraction of smooth muscle even after dephosphorylation. This is known as latch bridge phenomena. Very, very important. This is known as LATCH, latch bridge phenomena, especially in case of smooth muscle. This is known as latch bridge phenomena. So you have to remember, latch bridge phenomena is nothing but a sustained tonic contraction of the smooth muscle even after dephosphorylation. So whenever you are doing phosphorylation, dephosphorylation, you require the help of energy. But here, even after dephosphorylation, you are sustaining the contraction. That means I can understand latch bridge phenomena is a low energy utilized state, low energy but high tension condition of the smooth muscle. So the smooth muscle is generating high tension without using much of the energy. Low energy it is using because you have only causes dephosphorylation and phosphorylation once, but it is maintaining the sustained contraction. This way, smooth muscle uses less energy but maintains sustained contraction, and this is very, very beneficial for GI smooth muscle and other smooth muscle like gallbladder and all, because if it is utilizing huge amount of energy like that much of the skeletal muscle, then during GIT's movement, you require huge amount of metabolic rate, huge amount of basal metabolic rate. That way, this mechanism is very, very beneficial by which smooth muscle maintains its contraction for long duration, that is latch bridge phenomena. All of you know that in case of skeletal muscle, we have understood one phenomena, and that is known as the rigor mortis, right? Rigor mortis is a sustained contractile state in case of skeletal muscle, but that's a pathological condition. In case of smooth muscle, this latch bridge phenomena is absolutely physiological phenomena, right? So this is all about smooth muscle that you have to remember. So this is all about nerve muscle physiology.